Good morning, everyone. Thank you for tuning into this presentation today. My name is Prem Mohanty. I'm a product marketing manager and a life sciences product specialist at Benchling. I'm excited to talk about antibody-based immunotherapy R&D and some of the critical considerations in the associated data management. First, a little bit about Benchling. Benchling's mission is to accelerate life science innovation for the betterment of humanity. Our company was founded in 2012 by a team of scientists and engineers at MIT who were frustrated with the poor quality of software they had available to support their science. So, with the support of top tier investors like Anderson Horowitz, Thrive, and Benchmark, Benchling built out a range of solutions for life sciences on a single platform. We are fortunate to work with over 350 innovative life science companies, including many of the world's largest enterprises. The biopharma industry has evolved tremendously over the past decade. We are proud to work with several leading companies that have helped develop new advances in immunotherapy. We have been able to scale with these global biopharma leaders as they built out their immunotherapy portfolios. We are closely aligned with these leading innovators and work in partnership. This has helped us build a domain expertise in immunotherapy and antibody engineering. Over the past several decades, immunotherapy has grown significantly in terms of complexity. Today, it includes a wide range of technologies and methods. Some of the specific drivers of this complexity are: first, the antibody candidates being developed are just much more diverse structurally. In addition to well established monoclonal antibodies, we have a plethora of bispecific formats and antibody drug conjugates. Second, there is a greater emphasis on comprehensive characterization. As more candidates enter the market, the bar gets higher for follow on candidates. Finally, there is increasing reliance on lab automation to increase throughput, improve productivity, and achieve greater efficiencies. We have seen our customers align their efforts into scientific platforms. So it's not just focused on a single product, but instead they focus on a distinct pathway or technology capable of producing multiple therapies across multiple indications. As the science becomes more complex, we are seeing teams become much more specialized to increase the depth and efficiency that they bring to each step in the R&D process. Each of these teams is producing not just more data, but more complex data. There are more contextualizations that need to be maintained as you move downstream in the process and across programs. There are many more streams of data, from lab automation and manufacturing sensors to newer sources such as real-world data. So the industry is looking towards digital solutions here to not just manage the volume of data, but to bring order and structure to it as well. If we look specifically at typical antibody engineering teams, we see several common challenges. An antibody engineering scientist is dealing with archaic software tools that are highly manual and have low throughput. Additionally, capturing institution knowledge at the sequence level is often not possible. An antibody development scientist is dealing with cumbersome sample tracking both upstream and downstream and has to piece together information from multiple sources. And finally, an analytical scientist is struggling with handoffs of data, samples, and context between teams, which makes it really difficult to share learnings across program teams. We think every company and scientific team deserves a solution that will accelerate and support your innovations. When you think about the ideal traits that any digital solution should have in R&D, there are really four core attributes to consider. First, the built for complex science. It has to model the science you focus on, which means having the ability to build data models that fit your science. Second, adapts to your process. It has to support the processes and workflows that your team performs and have the ability to scale, iterate, and evolve over time. Third, it should enable key decisions. It has to empower users with data at every level, giving scientists insights about their experiments and team leaders data on their programs so you can make both scientific and operational decisions quickly with complete and accurate data. 4. Easy and intuitive to use. This ensures that users will immediately recognize the value of the solution, see that it saves them time, and adopt it in a way that will help keep them compliant and consistent with the rest of the R&D organization. Let's look at each of these attributes a little more closely. So what does it mean to be built for complex science? There has to be alignment between experimental workflow and the way that the underlying biological and chemical entities are modeled. As an example, we support a lot of customers who are working on the next generation of antibody therapeutics. They are mainly different. There are many different approaches here, from monoclonal antibodies to bispecifics to antibody drug conjugates. The science here generally involves the iteration of plasmid design to express and purify novel proteins, combined with in vitro and in vivo evaluation and optimization. While this is a very simple view of the process, it's actually very complex to model this. Every fragment of the antibody used has their own sequence and properties and ontology. Every resulting antibody has experimental results in vitro and in vivo that need to be tied back to the drug candidate and its underlying parts and sequences. So this is clearly a data modeling challenge, but it's also an operation challenge. Since these also represent the day-to-day functions for lab scientists developing these therapies. When we think of preparing digital infrastructure for these kind of challenges, we have to think about both the data management and structure as well as the operational considerations. The way we think about this is to build applications that follow the operational workflows, from in silico design to experimental execution, but to make sure the data capture flows into a structured data model as well. This means at any point in the process, you should be able to trace your work back to all the preceding steps and underlying sequences. And more importantly, for scientific decision support, the data can be readily queried across any dimension of the data model. This is a necessity to start building in automation and machine learning routines. So that's an example of how a very complex scientific process can be structured into a digital solution. But the hallmark of any good program is that the science will evolve over time. So what happens when things change? Digital solutions have to be adaptable. The IT world has moved away from monolith software towards more agile approaches. But adaptable shouldn't always have to mean coding and application development. Many life science companies either don't have the resources to do this or want to use the resources they have to focus on higher value operations. This is an illustrative example of a protein purification process, and it reflects the constant iteration and evolution that these specialized teams see on a daily basis. So in a real world, the data model, the process, and the workflows should be configurable in a low to no code environment. So companies can iterate and evolve quickly without being resource intensive or overly dependent on their digital partners. One way we seek to achieve this is with a configurable data model that is editable right from an admin panel. So users adjust the relationships and metadata on a particular entity quickly and without code. This also applies to process workflows, where you can update iterative processes like the example you see here. A modern digital solution should enable its users to make data-driven decisions. Different members of an organization have different questions that they need to answer and have varying decision support needs. For a scientist, this means providing ready insights across their experiments so that they can design, execute, interpret, and optimize their studies more efficiently without tedious data aggregation and data cleanup. For example, a scientist can compare the expression profile of multiple cell lines and antibodies across programs and studies by querying a central database where the data is already standardized during data capture. At the level of a team manager, the questions that need to be addressed are at the process and team productivity level, such as the ability to track outstanding requests, and the progress of related experiments. Finally, at the executive level, you want high-level view of your entire pipeline, the ability to compare candidates, to make no-go decisions, and to allocate R&D resources efficiently. Ease of use is the final most important attribute of a modern digital solution. Most software tools have a learning curve, and then because of poor user experience, a forgetting curve downwards as well. Only if the solution is easy to use can this ensure that users not only get trained, but ramp usage and realize the full value of the solution. I can share an example of one of Benchling's customers. Agenus is a publicly listed clinical stage immuno-oncology company based out of the Boston area. Prior to using Benchling, they operated from a variety of point solutions. This made it difficult for scientists to find information and collaborate, especially across multiple R&D sites in the US and EU. Additionally, because of the complexity of their biological workflows, it was difficult for them to keep track of their experiments and samples. They adopted the Benchling platform back in 2016 with the notebook, molecular biology, and registry applications. They have subsequently expanded to include the entire Benchling platform for their scientists. Their entire antibody research team can now collaborate better around a single unified platform with a complete view of their experiments, samples, and results. They can mine data across past experiments to optimize future studies as well. This is just one of many examples on how Benchling customers can accelerate their R&D using Benchling. And you can find many more of these success stories on our website. I would like to end on this slide about the Benchling solution. The Benchling Life Sciences R&D Cloud is designed to give you a complete view of your R&D innovation. Benchling's application spanned the R&D process from experimental design, in silico biology, sample tracking to process management. There are 7 applications that Benchling offers, but they're all offered from a single interface. This makes it easy for users to leverage what they need without leaving the software. All of these applications are built on a common unified platform. They share a configurable data model that maps to each team's unique scientific and operational requirements. Benchling is built on a modern cloud infrastructure and is fully managed by Benchling and offers state-of-the-art security and control. And we know that most companies have a rich ecosystem of instruments, software applications, and data stores that their R&D depends on. So we provide an open platform with full set of REST APIs and out-of-the-box connectors for lab automation, as well as ways to move data onto and out of our platform. Thank you for joining us today and for submitting questions. I hope you learned something new about Benchling and how we are helping organizations accelerate their immunotherapy R&D. Please reach out to me with any questions. Thank you once again for your attention.